Welcome to Bibliophil's e-classroom. In continuation of the previous lecture, in this lecture I am going to talk about the various stages involved in preparation of vaccines. So let's begin without wasting any further time. According to the CDC, which is an acronym for Centers for Disease Control and Prevention, is a national public health institute in the United States, there are 6 stages of a vaccine development. These are exploratory stage, preclinical stage, clinical developmental stage, regulatory review and approval stage, manufacturing stage, and quality control stage. So let's look into these stages in detail. First, it is exploratory stage. The exploratory stage is also known as the research and discovery stage. The stage involves basic laboratory research. Scientists identify natural and synthetic antigens that might help prevent or treat a disease. These antigens could include virus-like particles, weakened viruses or bacteria, weakened bacterial toxins, or other substances which is derived from pathogens. Then comes preclinical stage. This is where scientists start testing the vaccine in cell cultures and in animals to see if it triggers an immune response. If there is no immune response or the vaccine is causing harm to cells, then it is back to the exploratory stage. So the preclinical studies use tissue culture or cell culture systems and animal testing to assess the safety of the candidate vaccine and its immunogenicity or the ability to provoke an immune response. Animal subjects may include mice and monkeys. These studies give researchers an idea of the cellular responses they might expect in humans. They may also do challenge studies with the animals, which means that they vaccinate the animals and then try to infect them with the target pathogens. So all these are done to get a safe starting dose for the next phase of research as well as a safe method of administering the vaccine. Then comes the clinical development stage. After the preclinical stage, when we have a vaccine that looks both safe and promising and preventing infection in laboratory, we begin testing in humans because we don't know whether or not the vaccine will work. Then we begin a very structured set of studies called clinical trials. The clinical trials generally involves 3 phases. In phase 1, the experimental vaccine is given to a small group of people with the goal of determining whether the candidate vaccine is safe and to learn more about the responses it provokes among the test subjects. These studies involve healthy volunteers who are told in detail about how and why this vaccine is being tested and about any potential harm. This educational process is a process called informed consent and goes on before and during the trial. There is a continual discussion that happens between the researchers and the study volunteers, describing the study, informing the volunteers about any new information that happens at any time during the study, and also answering all the questions that the volunteers have. The study staff also determine whether the volunteer meets all the eligibility criteria to participate, which involves physical exams and blood tests. Phase 1 trials usually examine the safety of the candidate vaccine. It examines whether the vaccine causes any side effects or any tolerability, whether the injection hurt, whether or for how long it hurts. In this phase of studies, scientists are also gathering information about the body's immune response to the vaccine to see whether it is a promising approach to preventing the specific infection. If an experimental vaccine is found to be safe, it moves on to the phase 2. Phase 2 includes hundreds of human test subjects, which is aimed to deliver more information about safety, immunogenicity, immunization schedule, and dose size. During phase 2, we start what is called a randomized double-blinded clinical trial. This means that after volunteers enroll in a study, a computer randomly decides whether they will get the vaccine or whether they will get the placebo, which is usually just salt water. We do this just to be able to separate out the side effects that are due to the vaccine from those that are just happening to the people in the community at the same time, like the rate of cold symptoms or headaches. And we double blind, meaning neither the participants nor the study staff know who is getting which type of injection. This actually prevents both volunteers and the staff from over or underreporting symptoms. The participants are advised to tell about everything that is happening to them. So the phase 2 trials confirm the safety in a larger group of volunteers and focus on the best dose and the best timing of vaccination to produce the strongest immune response. The promising vaccines that are safe and generate a strong immune response move into the phase 3 trials. Then comes phase 3 trials. These are the largest trials which can include thousands or tens of thousands of test subjects. This trial continues to measure the safety, the effectiveness, and the rate of infection in the vaccine and placebo groups. This trial is particularly aimed to detect the rare side effects that sometimes may not appear in smaller groups. This will also tell us whether the vaccine is able to prevent the infection, and if so, whether it is effective in all the volunteers or only a subgroup like the young volunteers or the women volunteers, etc. In this trial, thousands of people, usually the people in the outbreak area, will receive the experimental vaccine. One way to speed up phase 3 trial is to do something called a human challenge trial. In this human challenge trial, the people are given the vaccine, but then the scientists intentionally infect them with the virus to see more quickly if the vaccine really works. It might sound like an extreme way to speed up vaccine development, but it has been done in many times before, including with cholera, dengue, and flu. Then comes the stage of regulatory review and approval. If a vaccine passes through all the 3 phases of clinical development, then the vaccine developer submits an application for license to the competent authority. And in respect of India, it is the DCGI, that is the Drugs Controller General of India, is the competent authority. DCGI is responsible for approval of licenses of vaccines, and this DCGI is a department of the Central Drugs Standard Control Organization, that is CDSCO, which is the Indian regulatory body for pharmaceuticals and medical devices, which is equivalent to the FDA of United States. Then, after the obtainment of these approvals, comes the manufacturing stage, where major drug manufacturers provide the infrastructure, personnel, and equipment necessary to create mass quantities of vaccines. And then these vaccines are distributed widely amongst the masses. Then, after the approval and distribution is completed, comes the stage of quality control. See, the approval and distribution is far from the end of the line, because a variety of systems to monitor the vaccines is adopted after they have been approved and distributed. These systems include phase 4 trials, the vaccine adverse event reporting system, and the vaccine safety data link. These systems are designed to monitor the performance, safety, and effectiveness of an approved vaccine. Through these processes, the stakeholders, that is the lab researchers, the policymakers, the medical professionals, monitor the vaccine, whether the vaccine is performing as anticipated. So these are the long stages involved in the development of vaccines, and typically it takes 10 to 15 years to develop a vaccine. And the fastest vaccine we have previously developed was for the mumps, and that took 4 years to develop. So it would be record breaking if we are able to develop the vaccine for COVID-19 by 2021. So that's all for the day. I will be sharing more information on vaccines in my subsequent videos. So thanks for watching.